Oh, I didn't notice you there. Hello, my name is Danielle and I'm here with UBC Paths to interview Dr. Bamji and her 2 graduate students. Let's get into it.
Hi everyone, my name is Saakshi and I'm a graduate student in the Bamji Lab.
And hi, I'm Andrew. I'm a PhD candidate also in the Bamji Lab, and we're going to take you on a tour of our lab space.
Awesome.
Here's where our lab space starts. So we're located in the Life Sciences Institute, where we have several labs sharing an open lab space. But each of these benches is devoted to kind of a certain lab task. So looking at performing plasmid cloning, so if we want to infect a cell or transfect a cell with a certain gene, we're able to produce those gene products here, and then we can insert them into cultured cells, and that allows us to identify the effects of different genes on various measures of cell function. Over here we've got our imaging suite. So you can see Nima here is performing some image analysis on a neuron that was grown on a culture dish. And in here we've got our confocal microscope. So this microscope uses lasers to produce images that are always perfectly in focus. So this allows us to image everything from single synapses and the molecules that are present there, all the way up to whole brain slices, and looking at the morphology of things like neurons, oligodendrocytes, and other glial cells in the brain. So this is where we label different proteins with antibodies that allow us to visualize them under the microscope. So when we study tissues in isolation, we want to make sure that we're doing that in as clean a way as possible. And so these are our cell culture cabinets. And so these allow us to manipulate cells in a very controlled manner so that we can really study like what happens at a specific genetic levels.
So on the computer over there is the cell that comes from all the efforts that you put in here and the culture that you do in this room. And then you finally get that end product where you see your beautiful cell, and then you can analyze accordingly.
Wow, that's so cool.
We can actually see.
Is there a reason that they're pink?
So the pink is just a pH indicator. It's
Okay,
so that we
awesome.
can just quickly open the incubator and see if we've got any sort of contamination. Kind of our lab space. So we've got a lot of different benches where people are performing a bunch of different things. So again, things from immunohistochemistry, working with RNA, DNA, working with protein products by looking at Western blots, which Sakshi's been doing. So we've got a lot of things going on in the lab, but this is where all the magic happens.
Wow.
Okay, so my name is Shernaz Bamji, and our lab primarily focuses on the cellular and molecular mechanisms underlying brain development and functioning. So it's pretty broad, but I cannot narrow it any further because we started off being really interested in how synapses are formed and remodeled. And as you might hear later on from Andrew, we've kind of expanded to understanding how not only how neurons communicate with one another, but also how well they function and how myelin wraps around neurons to actually promote signal transduction.
My name is Andrew Thompson. I'm a third-year PhD candidate here in the Bamji Lab, and my work primarily focuses on the role of these cells called oligodendrocytes, which are these cells that wrap themselves around neurons and form essentially an electrical insulating barrier that allows neurons to talk to one another over long distances.
Hi, my name is Saakshi Champaneria. I'm a grad student in the Bamji Lab, and I joined the lab back in 2023. As Shernaz and Andrew both mentioned, that our lab focuses on understanding the cellular and molecular mechanisms underlying brain formation, underlying the development of the brain. And our lab majorly is interested in one such post-translational modification called palmitoylation, and each of the graduate students work on different palmitoylating enzymes called the DHHC enzymes. So my work is mainly related on one such DHHC enzyme called DHHC21 and looking at its role in neuronal morphology, synaptic plasticity, and other neurodegenerative diseases.
Well, I have always been interested in the brain and only the brain. So really, way back in undergrad, I wanted to be a psychologist. But when I did some of the psych courses, I actually did not end up liking any of my psych courses, but ended up loving all my cell biology courses. And so I always knew that I was going to be exploring the brain at the level of cell biology and molecular biology. I think that one of the reasons that I actually do love studying the brain so much is because it's so complex, and there's actually no end to understanding, how we work, how we learn, we remember. And I think that this is kind of the most interesting things about us, is how do we do all the amazing things that we do every single day. And so that's constantly what's motivating me. So basically I come into this office and I sit in front of that computer and I just spend about 8 to 9 hours there and I do not move. That's kind of not very exciting, doesn't seem very exciting, but during the day I will do a whole bunch of things like have graduate students come inside and talk to me about experiments that worked, experiments that didn't work. I could be writing manuscript, I can be reviewing manuscripts. I also am the co-director of the Javad Moghfagian Center for Brain Health, so I can be doing a number of different things related to kind of running that particular center. There are student committees to go to, there are student theses to read. So basically every day is just filled with a myriad of different meetings, sometimes by Zoom, sometimes in person, and then just basically meeting graduate students. And of course, one of the things that I also do is teach, and I don't spend a lot of time teaching, but since I'm in the Faculty of Medicine, but teaching is a big component of what we do as well.
My day, very similar to Shernaz, is different every day. In that the places that we might be are very similar, whether it's at the lab bench here, or one of the microscopes around campus, or at the desk analyzing data. The type of tasks that we're performing are always changing based on the kind of questions that we're asking. So whether it's kind of sitting down in order to analyze like the thickness of individual myelin sheaths on axons, or to quantify the number of RNA transcripts in a given cell, or to understand how different cells are similar or different in the expression of different proteins or RNA. There's always different things to do. I also spend some of my time volunteering with some of the student committees here on campus. So part of the graduate program in neuroscience is run the Neuroscience Trainee Association, which is part of the graduate program in neuroscience, which puts on student events for neuroscience students. That's part of how I spend my time here at UBC.
My day kind of is quite similar to how Andrew's day is, because we are all grad students. Our day is kind of different every day, be it culturing neurons on Wednesdays, to just imaging on Thursdays and Fridays, to running Western blots throughout the week. So each day is kind of different for us based on the questions that we ask, as Andrew mentioned previously. But I kind of also take out little amount of time from our hectic schedules to kind of do extracurricular activities. And I'm also part of different clubs at UBC and try to help out with that. I kind of like teaching as well, so I'm part of I kind of am into the Rex mentorship program, where I try helping out the undergraduate students, getting who are interested into research. And this semester I'm also TAing with Shernaz, because I'm kind of interested into teaching aspects of this as well. So that is usually how our day is. The best part about being a grad student is the amount of knowledge that you gain out of it. It's just unending, unanswered questions, and the skill set that you develop while you're studying a particular kind of research, right? The skills that you develop, be it your communication skills, be it technical skills, be it your personal skills, you get to develop that a lot, and also to be able to contribute back to the society in some way or the other, just to help out as in the world as you're doing something and giving something back to the society. That is like the best part of being a graduate student. And I wouldn't say the worst part, there's no worst part about it, but the fear of failure is one thing that kind of, demotivates you at some point in your life, and the pressure to be able to, be the best at what you're doing. That is something that you eventually learn throughout your grad journey, and you overcome that fear as well.
Yeah, so the first part of the question, I think undergraduates should try, and even if you just think you're a little bit interested in research, try and get exposed to a research environment as soon as you can. I think something that happens all too often is students leave research experience until their fourth year, and then they have to go and do an honors thesis or a capstone project, and they're really overwhelmed by it because it's exposure to a whole bunch of new things, and also trying to figure out if they actually enjoy that when they're trying to figure out whether they should go to grad school or whether they should kind of continue in research. So my advice is pretty simple. It's try and get involved in research from your first year. If you can, volunteer in a lab, if you can find a summer position where you're applying for a scholarship and even getting paid for your work during the summer, I think that's the best way to do it. And then you'll kind of get an idea of, is research something that I enjoy? If you don't enjoy it, you don't have to push yourself to enjoy it. There are tons of professions in the world, and research isn't for everybody. And so the earlier you find that out, I think the better.
The thing is that there are a lot more students, undergraduate students, than labs, and that's just a fact. And so you might have, an amazing, resume. You might have an amazing transcript. So you've been doing really well at UBC, and yet you're not going to be able to find a lab, and that's going to be really discouraging. So the things to do to actually get into labs, I would try and contact and make as much contact as you can with the PI. Don't get disillusioned if you write. You have to write about 100 applications to get even one person to respond to you. Sometimes we don't even respond. We get that many applications. Don't be despondent. Keep pushing at it and persevering. Of course, as Andrew mentioned, that it would be fantastic if you got to find positions much earlier on. But the fact of the matter is that we don't also take first year and second year students in the lab, and that's because you just don't know that much enough to actually contribute to the lab. And so there are some labs that do take people like that. There are fly labs and worm labs where they do need someone just to come in and kind of do the grunt work, and so you can kind of volunteer in those kinds of labs. And the other thing is try and go to events like Pizza and Profs, or Student Undergraduate Association or whatever, where you have opportunities to meet the professor. Because if we get so many emails, but if you actually make some sort of connection with us, and then they'll say, hey, I'm going to write to you tomorrow and ask if I can be part of the lab. We'll be on the lookout for your email, etc. Otherwise, it's very, very hard to actually break through to us.
Yes, actually there was. There was a moment for me as well, and a mentor for me. So it was both. During my undergrad studies, I was working in this lab in my second year undergrad, I would say. It was again a neuroscience lab, and I was just doing an internship for 2 months. And during that time, the very first time that I looked at a neuron under the microscope was the first time I realized how beautiful the cell looks and how complex our brain is. Such a tiny, tiny cell can perform so many actions all at once. And that actually motivated me to look and delve deeper into the area of neuroscience. And it was kind of a turning point for me when I realized that, this is something that I want to pursue in my life. And this is something that motivates and excites me every day. So that was that. And the mentor, I would say, the one person who always has been the role model in my life is my father, because he is a doctor, and I've seen him operate on patients. I've seen him deal with different kinds of diseases in patients, and being able to, come out of it and navigate it so beautifully has always motivated me to, get into the field where I can help people by, be it in the field of research, be it in the field of clinical thing or setting or whatever. But that has actually shaped me to get into this field of research, and yeah. Yeah, Shernaz done.
So the one piece of advice that I would give people who think that they might be interested in research is obviously get into a lab early and see whether you are somebody who can handle failure. Because basically you need to be able to fail at least 60 to 70% I should say, of your life to be comfortable failing 60 to 70% of the time to be able to be a good researcher, I would say, because there's a lot of times where research does not go the way that you expected it to. You can have good runs, but there's always people, and both of them have experienced that where things are just not going very well. So I think a researcher should not be somebody who's just aced every single science course that they have ever done, because that's a very, very different skill set. And being a researcher, you have to have a different skill set, and that would include perseverance, being very, very meticulous, and being a critical thinker. So not just memorizing things off a page, but by really kind of being more like an investigator.
I think my answer is kind of embedded in Shernaz's a little bit, so I'll just kind of emphasize maybe. But my answer would be organize. Make sure that your life is well planned out, so that when you're going through your day, you're not really thinking about, what should I be doing right now? Because that's just time that you could be doing something else, rather than just wasting it on thinking about what you could be doing. So that would be my one piece of advice. Whether you need a written down calendar or your calendar in your phone or whatever method you need to develop that works for you, find that as soon as you can and stick to it.